In this video we will discuss about DNA Sanger sequencing method. So from this figure, that is figure on the left side, we will understand what is the principle behind this DNA Sanger sequencing method. So this is the deoxyribose. It is usually the nucleotide that is present in the DNA, and because of this presence of this OH bond, it helps for the further extension of the DNA sequencing. It will bind to the another nucleotide. However, when the nucleotide is present as a dideoxyribose, then instead of having a OH bond, it only contains the hydrogen bond. And because of this, what happens is that it will prevent further extension of the DNA sequencing. So further extension of the DNA sequencing will not happen, and by analyzing the by analyzing with the help of gel, we can analyze the length of this DNA sequence. At the same time, we are also aware about the which deoxyribose nuclease terminates the sequence. We have that information. So based on these two information, we will be able to detect or identify the uh, sequence of the DNA. As it is shown here, a preliminary examples is that the G is present if the DNA the what happens is if the extended DNA terminates at this G point, and it is at 4 nucleotide position, then we can understand that the complementary position at the 4 nucleotide after the primer is the C, or is the cytosine. So this is one of the examples that is shown here. However, we will understand how it is actually been carried out in the lab or during the experiment. So we will have 4 separate solutions. Each contains all the 4 nucleotides, and simultaneously they also contain one of the deoxyribonuclease deoxyribose nucleotide. As in this case, what happens is that uh, T deoxyribose nucleotide is present, and this A deoxyribose nucleotide is present, this G deoxyribose nucleotide is present, and this C deoxyribose nucleotide is present. And this is the example that is considered. This is the DNA. This is the 5' to 3' DNA sequence, and what happens is that this DNA sequence has been added into each of these flasks along with the primer, and the uh, DNA elongation reaction has been carried out at appropriate temperature. After that, considered in this case one example. This is the T deoxyribose. So what happens is that one of the cases that it will form a uh, T at the first one of the condition is that T will binds to the first part of the A, and it will not further proceed. And another case is that the normal nucleotides will binds to the sequencing, and T will binds at the second at this at the end of this second primer. So in this case, we will be able to get two products from this DDTTP that are of our interest. So simultaneously, we will get other products from the other flask, and when we will run these gels on, when we will run these solutions on the gel, we will be able to detect the length of the sequence with the help of a distance of the band that is occurred from the starting position, and also compare them with the other bands. So one of the examples that we can see is that T is at the bottom. So so we know that the smaller the fragments, it can travel the fastest. This is the smaller fragment, so and it is found in the T. So the complementary sequence starts with T and then is C, G, A, G, T, C, A, G. So this is the complementary sequence that we get from this band, and when we get the complementary sequence we can find the actual sequence by just using the reverse analogy, that is G is binds to C and A is binds to T. That's how we will identify the actual DNA sequence from the DNA Sanger sequencing method. So however, the problem with this method is that it cannot identify a very long DNA sequence, as it will give a lots of error. So for the longer DNA sequencing, we will use different methods such as next generation sequencing. So I hope you have understood something about this DNA Sanger sequencing. If you need to understand this DNA Sanger sequencing in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.